Sharing presentation mode. Okay, thank you very much. So today I'm going to talk to you about our program looking to develop MAP4K4 inhibitors for the suppression of cardiac muscle cell death. So I'm going to start by giving a bit of brief rationale for our interest in MAP4K4, talk about the approach that we took to identifying suitable MAP4K4 inhibitors, and then finish by talking about some of the challenges associated with developing cardioprotective agents, particularly in the context of myocardial infarction. So this model, this program, as Alan alluded to, was a multi-center collaboration between ourselves largely and Professor Schneider's lab at the National Heart and Lung Institute at Imperial College. Okay, so MAP4K4 is a serine threonine kinase. It's an upstream member of the MAPK superfamily, and it's activated in cardiomyocytes on induction of cell stress caused by a variety of stimuli, including oxidative stress, and therefore is activated in oxidative reperfusion during myocardial infarction, and also is activated by a number of anti-cancer drugs with cytotoxicity, such as doxorubicin. So cardiac muscle cell death plays an instrumental role in both acute and chronic heart failure, and therefore small molecules that are able to protect cardiomyocytes have a potential therapeutic benefit. So this slide is just summarizing some of the published work from Michael's team in conjunction with ourselves, showing that if you knock down MAP4K4 with either genetic inhibition or inhibit it with small molecules, you're able to protect cardiomyocytes from a variety of death-inducing stimuli. So in this middle panel here, we've got effects of showing in the black bars that if you treat human pluripotent stem cells with hydrogen peroxide, you get an increase in cell death. And this can be prevented by shRNA targeting MAP4K4. In the upper panel, we've got similar experiments looking with small molecules, showing that small molecules are able to protect human pluripotent stem cells from hydrogen peroxide-induced cell death. And then the bottom right-hand corner, we've got some work showing that with doxorubicin, in this case in rat cardiomyocytes, but we've similarly shown this in human cardiomyocytes. And human and rat MAP4K4 have a high degree of homology. So the aim of our program essentially was, our main focus was on looking to try and show that proof of concept for small molecule MAP4K4 inhibitors in myocardial infarction. And to that end, the aim was to do some small mammal and large mammal efficacy studies with MAP4K4 inhibitors, and with the aim of targeting for the large mammal studies and also towards the clinic, we were looking to target an agent which is suitable for IV administration and therefore rapid onset. So this essentially means high aqueous solubility compounds with modest clearance and volume distribution. However, it can be difficult to maintain levels by infusion dosing in mouse for mouse studies. So we also had to bear in mind that we may need to deliver the compound orally or via IP in our mouse studies. So this here just shows the sort of screening cascade that we put in place. So as you would imagine, we had a biochemical assay looking at MAP4K4 and some kinase selectivity panels. And the primary cellular assays that we used on this project were looking at cardioprotective readouts against both the rat H9C2 cells and the human pluripotent stem cells with a variety of stimuli, including hydrogen peroxide and menadione, and also doxorubicin. In practice, these assays showed similar results across the board, and therefore we used the DOX H9C2 assay as our routine screening assay due to the more high throughput nature of this assay. Compounds were substantially optimized in terms of DMPK, etc., with the aim of selecting compounds for the small and large mammal efficacy studies. So the starting part for this program was the molecule shown here, F1386, which was identified following virtual screen. As you can see, this compound has reasonable MAP4K4 potency and excellent MAP4K selectivity. So then the right-hand side here, we've got a heat map showing the activity of MAP4K4 against a panel of 140 kinases, and only MINC1, which is approximately 90% homologous with MAP4K4, showed similar levels of activity to MAP4K4. A handful of other kinases had some activity, but it was a window of about 10-fold. However, this compound has poor DMPK properties. So very high microsomal clearance and poor aqueous solubility, and therefore wasn't suitable as an in vivo tool. So the aim really was to use the available structural information to try and generate compounds initially suitable for proof of concept studies and then for further development. So MAP4K4 has a tyrosine residue in the P loop, and this enables it with certain inhibitors to adopt a folded P loop conformation in which the tyrosine residue, tyrosine 36, is able to form pi stacking interactions with the inhibitor, and this can give rise to highly selective inhibitors. So this here is a model of F13A6 bound to MAP4K4. This has subsequently been confirmed with later ligands via X-ray crystallography. And the ligand adopts a propeller-like conformation in which the aromatic rings form numerous pi stacking interactions with tyrosine 36. The pyridine motif forms hinge binding interaction with the hinge of MAP4K4. To provide a bit of perspective here, we're just showing on this diagram here, the vector out towards the solvent. And this is largely where I will talk about today, which is our modifications in this region, which led to the proof of concept compounds. So the feeling was that in addition to being able to use this position to modify the properties of the molecules, we could also use this position to pick up further interactions by better filling out the space and also by picking up potential hydrogen bond interactions with the side chain of cysteine 108 and also with the backbone of cysteine 108 and the side chain of aspartic acid 115. So before beginning to optimize the molecules, what we did was a little bit of work looking to see whether we could replace the phenyl rings with heterocycles and therefore identify which was the most suitable cause for further elaboration. So to that extent, you can replace the phenyl ring in the front pocket with either 4-pyridyl or 3-pyrazyl groups. In the case of and these show slightly improved potency. In the case of the 3-pyrazyl group, this did, however, result in a slight reduction in selectivity. The 3-pyridyl compound was roughly equipotent with the phenyl compound. However, we decided to go forward for most of our compounds, largely with the 3-pyridyl and the phenyl compound, as we felt that these had the best vector for access to the solvent, to pick up further interactions and also to improve the properties of our inhibitors. In the back pocket, phenyl was the most potent of the small set we looked at. However, 3-pyridyl and 4-pyridyl also had reasonable levels of activity, and this was more potent than the 3-methylpyrazole. Again, we selected two groups, the phenyl and the 3-pyridyl, to go forward with. The 3-pyridyl compounds typically showed some improvements in metabolic stability. Having identified some cause on which to elaborate, the next stage was to try and optimize interactions with the water-accessible channel and also establish some linkers for appending some solubilizing groups. So to that extent, you can see in this right box here are some examples of compounds that were made, which showed high levels of MAP4K4 inhibitor activity. So these compounds were all sub-10 nanomolar in our MAP4K4 assay. So we could place an amide group into this position here, and we have some crystallography data showing that the amide is able to pick up some water-mediated interactions with aspartic acid 115. You can cyclize this round as well. It gives you highly potent compounds and introduce a further nitrogen atom, which can be subsequently elaborated to modify the properties of the inhibitor. Likewise, an ether compound gave highly potent inhibitors, which you can extend out to put a side chain on to subsequently modify the properties, etc. So this is just showing you some data associated with the compounds. So you can see here that the compounds are highly potent MAP4K4 inhibitors. They show good cardioprotective effects against the H9C2 assay. So here we've got EC50s and also the magnitude of the response, which is relative to this compound here, 5804. So we've standardized the maximum response relative to 5804, so you can see how each compound responds and show that they have a good cardioprotective effects. The compounds also had good microsomal stability, and pleasingly, they also had reasonable mouse PK. So at this stage, we thought we may well already have some compounds that might have potential to be used in proof of concept studies in mice, either by PO or IP dosing. However, even after formulation, these compounds, we could really only get solubility of about 100 micrograms per mil. So we didn't really have sufficient solubility infusion dosing for large mammal studies. So that end, where I guess we went down the direction that many a kinase program has gone, is looking to append solubilizing groups to the inhibitor, and you can use the AMAD or the ether chain to append a basic solubilizing group. And by doing this, you can get some significant improvements in formulated solubility. So you can now get compounds that are 1 to 2 mg per mil soluble. What we did observe was a loss of cellular protection. And we think this might have been due to the introduction of cytotoxic effects due to the lipophilic basic nature of compounds. As you can imagine, with small molecules, particularly kinases, inhibitors, you're usually looking to kill cells rather than have a cytoprotective effect. So we went back to revisit the neutral compounds and look to see how we might be able to improve the solubility of these compounds. So to that extent, we decided to investigate some prodrug approaches. So we took 2 different prodrug approaches to offer the maximum amount of versatility. So the first prodrug approach, which I've labeled type A, was to apply a methyl phosphate group to the pyridine motif, which can be hydrolyzed to give a hemiaminal and then release the parent drug molecule. The second approach was to introduce an alcohol side chain into the group, pointing out to the solvent, which again could be used to generate a phosphate. At this stage, we felt we had molecules that might be suitable for a type A prodrug approach, but we didn't have any molecules suitable for a type B prodrug approach. So we decided to make some molecules that were suitable for a type B approach so that we could compare the 2 approaches side by side. Pleasingly, you can introduce alcohols into the side chain and retain high levels of MAP4K4 activity. However, in the case of the primary alcohols, these were highly metabolized by mouse and human hepatocytes. You can improve the metabolic stability by adding a single methyl group. However, typically you need to add 2 substituents. So this can be done in the beta position, as exemplified by this oxidate here. You can also put cyclopropyl and dimethyl here, and this gives some improvement in metabolic stability. However, the most improvements in metabolic stability were obtained by putting 2 groups alpha to the hydroxyl group. So again, just showing you some activity associated with these compounds. So again, we have potent MAP4K4 inhibitors. We should have good EC50s in the H9C2 assay and good overall responses in terms of the cardioprotection. The compound showed good hepatic stability, and again we were able to obtain good mouse PK. And so, once again, we felt from this approach we had compounds that were suitable, potentially suitable for mouse efficacy studies. So at this stage, we decided to take a molecule which we called DMX5/4, which might be suitable for a type A prodrug approach, and take a molecule 9117, which might be suitable for type B prodrug approach, and compare these side by side. So you can see here that we have compounds have both got good MAP4K4 potency. They show good protection in the H9C2 DOX assay, and the more relevant assay from the point of view of reperfusion energy, hydrogen peroxide protection in human pluripotent stem cells. Again, the compounds show good inhibition and good magnitude of response. Pleasingly, you can dose these compounds orally to mice and get good free drug levels equivalent to the sort of levels that you are required for inhibition or cardioprotection in the in vitro assays. And this just here shows you some of the efficacy data that we've generated. So the y-axis shows the infarct size relative to the area at risk, which is a standard way of normalizing the infarct size. But you can see here for the DMSO control group relative to the 2 compound treated groups in the mouse in vivo model, that we were able to get at least 50% reduction in infarct size. And this was dosed 1 hour post induction of the infarct. So the next stage was to convert these 2 molecules to the corresponding prodrugs. So DMX5/4 was converted to DMX10001, and DMX9917 was converted to 12213. And to evaluate these with some in vivo PK experiments to show that the compounds could be rapidly cleaved to the corresponding parent molecules. So at the right here is just showing the data for DMX10001 in rats. And you can see the blue line here shows that we get rapid disappearance of 10001 and rapid appearance of DMX5/4, showing that these compounds do indeed act as suitable prodrugs. And we have similar data for 12213 which I've not shown here, and we've now shown this across multiple species. So now I've established that both approaches in principle could work. The next stage was to select one of these two molecules to take forward into a large mammal efficacy study. So to do that, what we made was crystalline sodium salts of both compounds and assessed their solubility. And what we found was that the prodrug DMX-10001 was approximately 10 times more soluble than the prodrug 12213. And we assign that essentially to the removal of the amide, which might weaken interactions within the crystal lattice. In fact, we have made a prodrug of 9917 of this position as well, and that is equally soluble to the prodrug 10001. So 10,001 was selected for the large mammal efficacy studies. As we've removed the hinge binding units, this compound essentially has no kinase activity, and the parent molecule DMX-5884 is a highly selective MAP4K4 inhibitor. So in a panel of 376 kinases, it only really inhibits MINC1 and TNIC to similar activity, and these two kinases have 90% homology to MAP4K4. There are a small number of other kinases which are inhibited with approximately a 30-fold window. So the next stage was to progress this to porcine efficacy models. So the first thing to do was to establish an infusion regime which would enable us to get enough compound on board. And this red line here is showing the free drug levels we were able to achieve following the infusion dosing. So we were able to achieve an average free drug concentration of just over a micromolar. And in black here is showing the sort of free drug concentrations which were achieved in the mouse efficacy study, with an average concentration of about 100 nanomolar. So in the pig, we were able to achieve at least tenfold the levels that we had shown efficacy in the mouse study. Also, it's worth noting that this sort of level of 100, about 1 micromolar, is at least threefold the sort of typical free drug EC50s we were observing in the cell assay. So we felt at this stage that we obviously had enough compound on board, and we were able to achieve our sort of medicinal chemistry objectives to try out a sort of pig efficacy study. So compound was scaled up, and unfortunately, we only observed a small reduction in infarct size relative to the area at risk in this study. And I guess this, although obviously very disappointing, this does allude to some of the challenges associated with trying to develop cardioprotective agents, particularly in the context of myocardial infarction, some of which I'll just touch upon on my last slide. So, as mentioned, there are a number of challenges in translating this sort of work, into clinical trials. And one of the key things is the need to do large mammal efficacy studies, which are considered the gold standard. And although these show much better representation to humans, it can still be challenging to replicate human infarcts. So, for example, in this study, we had infarct size relative to the area risk about 80% of the control groups. And it's known that obviously the larger the infarct, the more challenging it can be to provide cardioprotection. And typically the medium size of human infarcts are 50 to 60%. So there's across studies, there's lots of studies in the literature, and they will have different infarct size depending on the species of pig, the exact protocols, etc., the group that have been used. So it can be obviously sometimes challenging to compare studies. The other challenge is really trying to get a handle in the early stages of drug discovery program about whether the compounds are likely to work. For this, we've used the human pluripotent stem cell cardiomyocytes, which have been used mostly from the perspective of looking at cardiotoxicity. But again, here at the moment, there's obviously no standardized way of looking at this. The choice and strength of insult can affect the level of protection that you can achieve and the potency of your compounds. So these are some of the challenges that that have to be worked out if you're looking for cardioprotective agents in the context of myocardial infarction. So finally, I'd just like to thank all the people that worked upon with this project. Particularly Michael Schneider's group at the National Heart and Lung Institute, also Dirk Dunker's team who did the large mammal studies, my former colleagues at DomainX and Wellcome Trust and Apollo for the funding, and you for listening. more than happy to take any questions. And you can also email myself or Domainix if you have any further questions about the project. Thank you.